So thank you very much. Then I guess I'll get started. So I will start. So for my experiment, functional, basically what I am looking at here is functional genomic studies. Typically uses spatial analysis for these comprehensive and description and discovery of these gene expressions. So tools such as, let's say, PGC, polar gene curve, which is a tool that helps generate these comparisons of gene expressions with spatial data. However, these tools do struggle with low coverage genes because as the coverage decreases, the gene do express distributions among cells. They do become sparser. So with that, I would like to introduce to you guys in this session radar scanning plot method, which is an alternative method that addresses these limitations basically through scanning sectors inside, angled sectors inside a whole t-SNE plot. So let's delve into this. So the motivation from this comes from the basic t-SNE data embedding techniques, basic common embedding techniques, where we try to map these high-dimensional cellular data into hopefully 2D or 3D plots. So basic techniques involve preserving distances, such as t-SNE, mapping these distances into a more of a 2D understandable version of the whole data. However, there are a few additional properties that we need to consider other than the position of cells inside a particular cluster. For example, the first one being coverage, specifically coverage in terms of bias. So as you can see here, in this particular cell, it is expressed in both clusters, yet in this cluster, the neutrophil, this cluster, this gene actually expresses a particular bias towards the bottom, while in the other section, in the other cluster, being monocytes, this one is covered basically for the whole cluster. The other one that we have to consider is density of a cell in a cluster, particularly when it deals with dense distribution, or whether it is more of a sparse distribution around the edges. The final thing we have to consider for cellular data is its cluster shape, or a particular shape of marker gene within a cluster, being whether it is symmetric or non-symmetric, as shown in the figure. Okay. So the challenge starts with how we can characterize these non-uniform distributions of genes, of cells expressing a certain marker gene. So in the example I referred to previously, we have the neutrophil being this cluster and the monocyte/macrophages being the other. So as mentioned before, we have three cases where we have an issue of, for example, this gene being present in only one of the two clusters. The second being that a gene is present in both clusters, yet one of the clusters shows a significant bias compared to the others in terms of coverage. And then finally, we have a case where it is present in both clusters and more, but in the monocyte cluster, it shows certain phases where the coverage becomes increasingly dense. So let me introduce one of the previous methods developed. It is called polar gene curve, where it aims to separate a certain cluster into marker genes, which is the foreground of the whole cluster, as well as the rest of the cells that do not exhibit the marker gene, known as the background. So what the polar gene method does is it splits it into a 360-degree scan, where each angle will be represented by a foreground histogram and a background histogram of projections, and then it calculates the Gini index, which is basically an economic thing, and then which serves as a good criteria to plot out the differences between the foreground and background. However, there is a potential weakness of PGC because with all calculations, there is this possibility that with a lower coverage, you will get potential noise, and as well as some instances where you might have certain genes with a high coverage significantly impacting the final PGC figure, as it is very sensitive at a low coverage. So you can see here that with the high coverage, it performs really well, as it pretty much aligns with the whole background. And then, but with a coverage such as like 15%, you'll start to see these random, unnecessary spikes begin to pop up that might not be the case for when you look at the actual spatial data. So I introduce RSP. We start with a similar approach that polar Gini curve originally starts with, which is to isolate the cells expressing the marker gene from the background. So we can see here that, for example, in this neonatal mouse heart simulation data set, you have the background being the cells in gray, and then the marker genes being colored in from dark blue to yellow, being the expression levels from 0 to 1 normalized. Then we introduce the idea that we divide these cells into angled bins, and then we characterize basically into these foreground and background distributions, and then finally we calculate the difference. So over here is a simulation of 1,000 points inside a unit circle, where we highlight 50% of this marker cells just to show what it might look like with this. So you can see here that we have divided this whole figure up into 100 angles, and then each angle will be considered as a bin to be plotted with the histograms. So at each particular angle, let's just take 0 degree for example, what you'll get is 2 histograms. You'll have the cell angular distributions with the foreground being displayed here as red, and then the background being displayed as blue. The theta degree is defaulted to from 0 to 2 pi, which is 360 degrees. However, the method does allow you to change the different theta bounds so that you can see maybe different sectors of the actual polar plot. And then the delta, the change in theta is calculated basically based on resolution, which is a parameter in the method that basically determines how many bins there are. We have defaulted it to 1,000, as we have noticed the figure to be very smooth. So yeah, we have noticed that with RSP, with radar scanning plot, we have produced 4 types of visualizations that are indicative of cell coverage. So over here, we have the first one in which we have an even distribution and then a high cell coverage. So in that case, you will notice almost negligible area with the RSP polygon. Additionally, with the lower cell coverage, yet with even distribution, what you'll see instead of random spikes is it is more detailed. It has a more defined shape, showing potentially where the cells exactly is compared to PGC, which is just a smooth line pinpointing at a general direction. For bias distributions, which are these 2, you'll see that with a lower cell coverage, you'll see a bigger shape. This would indicate that with sparse data, yet with a biased population, you'll see generally where it is biased towards in the angle. And then for high cell coverage, you'll see a similar shape compared to the even, but of course, you'll still see the direction where it's biased towards. So to go into the method in more detail, after we generate foreground and background, after we generate the foreground and background distributions for each angle, what you'll get is a 360-degree representation of this figure. To get a numerical value for each of the radius plot over here, we just took the foreground in the cumulative distribution curve. So this is the histogram. Once we made it into a cumulative, then we can find the absolute difference in this and then get the radius based on the difference against the total expressing cells in the cluster. So this step is mainly to normalize it so that it can be between 0 and 1. So comparing RSP radar scattering plot to polar Gini curve method, as we have noticed, it reveals distinct patterns that are not really identifiable with PGC. As you can see here, with a simulation gene coverage of 5%, we can see here that RSP have a significantly more defined shape when it comes to identifying where they are expressing cells with the marker gene, instead of PGC just showing a spike in a general direction. Additionally, when we're looking at the accuracy, this plot over here shows the RMSD of the polar Gini against the coverage, and the panel on the right shows the RSP area versus the coverage. So we can see here that compared to the polar Gini's curves, higher variability in that lower coverage range, there is a consistent decrease over here, which indicates that it is more reliable when it comes to looking at coverages like these. The other concept is that basically looking at coverage of these particular genes inside the RSP area, we can see that there is a log decrease from the lower coverage down to the higher coverage, which shows that as coverage gets bigger, you will see a decreasing level of RSP areas. With these larger areas of genes and various coverage over here, you will tend to see them falling into the other 3 types of genes that we have previously noted over here. So type 2, 3, and 4 are these 3. So depending on the particular threshold depending on the expression data you want to input, you will get different representations, different categorizations of the 3 types. So in a real-world case study, we have conducted a reanalysis of the mouse neonatal heart dataset, and we have found, based on the RSP area, we have found multiple genes that are highly expressed, and as well as potential marker genes for future revisions. For example, we have genes like SPARC, which are really commonly expressed in a lot of the cells and are responsible for really development purposes. Additionally, we have FABP4, which is directly related to heart failure, and it is related to transport fatty acids and etc. GPX3, it is related to, negatively correlated to fibrosis, and does have a suggested role in cardiac tissues response and remodeling. So these are all type 1 genes. Now for type 2 genes, you can really see that RSP have identified multiple genes that are responsible, that have a significant role in the neonatal heart. For example, in a mouse neonatal heart, UQCRFS1 is really important because it handles ATP production in mitochondria and such, like NA38, ATP6V1F, and etc. These are all genes that do have relationships with either hearts or neonatal development. Finally, RSP, because it is so much better at handling lower coverage genes, there are potential indicators for co-regularization. For example, for these two genes, which are related to collagen type 1, these genes do demonstrate complementary distribution patterns. For example, you can see here that over here, the bias is towards this part, while the RSP plot for the marker gene collagen type 1A2 is pointing in the other way. So it has proven successfully in identifying these gene pairs as per the previous analysis, and then we hope that this opens up new avenues for the discovery of possible gene pairs with this robust tool. So yeah, thank you very much.
Thank you, Peter. Are there any questions from the side of the audience? If this is not the case, maybe I can ask you, did the concept of this radar scanning plot have any, let's say, type of biological motivation? Because any type of data construction is associated with information loss. Yeah, and let's say, is there any biological idea behind that type of data projection methodology that you applied? Or is it just the idea that, let's say, you concentrated data from certain sparse regions? Or
So the motivation behind the RSP method derived principally from the polar Gini curve method, where you are dividing essentially a cluster data into these angled bins, let's suppose. And so compared to PGC, which is quite computationally inefficient due to the thousands of experimentations that it actually carries out, I would say that the principal motivation is more of a computational improvement. But biologically speaking, it does opens up. It's more accessible because it both incorporates the RSP area, which is a numerical value that you can take to find potential marker genes, as well as conducting qualitative analysis on the actual figure itself, so that you can see where it's biased towards more easily with the whole data being imprinted on a radial plot.
Okay. Let's say I'm asking this because, without taking the specific mathematical formalities into account, imagine if you have such a projection from a center, and on one line that goes through the center, two of your data points are located. They will be projected easily to the same dot. So regardless to which type of clusters they might belong, regardless of their actual distance along that line,
can I address your question?
Please,
so I think the motivation biologically is that our current single-cell analysis is very crude. Right? So we only just, actually, as Soren mentioned in the previous Q&A sessions, and biologists are just interested in cell type and then what genes are abundantly expressed in the cell type and move on. But as Peter already presented, that there are so many different shapes and forms. If you actually use the gene expression, not just one as a thresholding way, you'll find many interesting distribution that indicating that these clusters that we define can be subclustered. Maybe biologically it means that certain cells have different phases and certain cells have different, like B cell at naive and then mature or depleted or B cell T cells. So all of that may be actually discovered with a method like this. If you find the marker genes and thresholding it and then have all different kinds of subdividing possibility and also being able to find cell type specific gene co-regulation patterns.
Okay, thank